friends, we were discussing about various factors that affect the growth of hairy root cultures in bioreactors, and in that we started with aggregation and adhesion of the cells. And we discussed that if the aggregates of the hairy roots is in the size range of 2 to 10 millimeter, then they lead to the inhibition of oxygen circulation in the media, they inhibit the transfer of nutrients into the media, and eventually the innermost cell, they lose their viability, which eventually decreases the overall secondary metabolite production. And in order to prevent those aggregation, if we use high speed impellers or high speed bubbling of air into the media, that also leads to the adhesion of cells onto the walls of the bioreactor, where eventually the aggregates develop over the period of time, which also develops the senescent patches into particular culture of the hairy roots. But we, on the very same side, we also discussed that aggregates are important because they allow cell to cell communication. They help in maintaining the viability of the cells, because if single cells are present or very small aggregates are present, less than 0.5 millimeters, then these smaller aggregates or single cell also lose their viability. So aggregates are useful, but up to a particular threshold size. Beyond that, they hampers the growth of the hairy roots. Now, how we can prevent the formation of larger aggregates? Now, there is another problem with large aggregates. They tend to sediment. So therefore, we always try to maintain the size of aggregates in the range of 1 to 2 millimeters and not more than 2 millimeters, with a specific gravity of 1.002 to 1.02. Now such size of aggregates or aggregates of such size, they keep on floating or suspended into the media, they don't get sediment. We can also prevent the formation of large aggregates by adding pectinase enzyme. Now, as we discussed that cells produce certain polysaccharides which help in adhesion of these cells, and one of such polysaccharide is pectin. Now, these pectinase enzymes basically they degrade that pectin, making the cells free from each other. So, this is one approach. You can add pectinase enzyme into the media which dissolves or degrades the pectin, thereby preventing the formation of aggregate. Second is we can also add polyvinyl pyrrolidine into the media. Now, this agent PVP, it forms a film around the cell, thereby preventing the aggregation, preventing the formation of large aggregates. Now, it has been seen that calcium ions, they form complex with those pectin polysaccharides, thereby hardening the joint between the two cells, thereby eventually leading to the formation of larger aggregates. So, it has also been found that if we reduce the calcium ion concentration in the media by reducing the addition of calcium chloride into the culture media, this also weakens the adhesive material that is forming between the two cells, and eventually it helps in preventing the formation of large aggregates. So, these are the few approaches by which you can reduce the aggregation of hairy root cells in the culture. The second important factor is mixing and viscosity of the media. Now, we know that mixing of the particular culture media is important because it helps in proper transferring of the nutrients. It is helpful in proper circulation of oxygen across the media, thereby allowing the cells to grow, and it also helps in prevention of the aggregation. But again, mixing at a higher speed than the required will have a negative impact, because it will lead to increase in the shear stress onto the hairy root cells, which eventually lead to the lesser production of the secondary metabolites or lesser growth of the hairy root cultures. And too slow of mixing speed will allow the cells to form aggregates, thereby leading to all those problems which we have already discussed. By and large, it has been found that mixing at a rate of 100 to 150 ppm is suitable for hairy root cultures, because these cells are much larger in comparison to the microbial cultures, and these cells can bear the shear stress induced by mixing better than the microbial cultures, wherein the mixing speed is quite low. Therefore, most optimum speed has been found to 100 to 150 rpm in a particular bioreactor. Now coming on to the viscosity part. Now it has been found that the plant culture media or plant culture along with the media has a non-Newtonian rheological pattern. When we talk about this kind of rheological pattern, it means that the viscosity of that particular media is not constant and it changes with the application of shear stress, or it changes with any other factor that affects the viscosity. Now it has been found that the overall volume of the bioreactor, around 40 to 60% of the total volume, is occupied by the cells growing in that particular culture. So when this kind of volume is occupied by the cells, they definitely will affect the viscosity of that particular media, and eventually they will change the viscosity. So we can say that viscosity of a culture media, it changes with the increase in cellular density, and it increases tremendously, steeply, if the cellular density increases beyond 10 gram per liter concentration. And once the cellular density exceeds the prescribed limit or the threshold value, then there is a significant increase in the viscosity, and this increase in viscosity will again inhibit the proper circulation of oxygen. This will also allow the formation of aggregates, because now the roots, since they have a very intricate growth, these roots will they will come in contact with each other, because the shearing or the rotation or the mixing will not have that much impact if viscosity is very high. So therefore, these roots will become more stronger aggregates. They will inhibit the flow of oxygen, they will inhibit the exchange of nutrients. This will eventually lead to the decrease in the production of secondary metabolites and polysaccharides. Now it has also been seen that the production of secondary metabolites and polysaccharides by the cells also increases the viscosity of the media. So we can say all these factors, they are responsible for increase in the viscosity, and the increased viscosity has a negative impact on the growth of the hairy root cultures. So these are some of the problems which I have already discussed, that there is an improper heat or mass transfer in the media if viscosity is increased. Then there will be non-uniform temperature across the culture media, because where there are dense aggregates, the heat transfer from the central core will not be proper, so they will form cooler regions in comparison to the outer portions where there is a proper heat transfer. Then pH, again pH will be, because we know that as the media get exhausted, its pH drops. So from the in the central core of the aggregates, the pH will be different. Then oxygen concentration, obviously we have discussed, there will not proper flow of oxygen. There will be the formation of dead pockets where the oxygen flow is not there. There is not proper flow of nutrients in that. Those areas will become dead pockets, and senescent patches will develop in that particular area, and there are increased chances of aggregation. Now the third factor is aeration and shear effects. Now we know that since cells are growing in a media, they require oxygen. So the demand of oxygen is high for such cells, and it has been found that the concentration of 1 to 3 millimoles of oxygen per gram per hour is sufficient for the proper growth. Now aeration has been found to be done at an appropriate rate. Why? Because if the aeration is very slow, again it will lead to the decreased oxygen concentration in the media, and that will develop certain dead pockets into the media. That eventually will lead to improper or non-uniform growth of the media. If the aeration rate is too fast, it will lead to the formation of foam in the media or large bubbles in the media. And we have discussed that if bubbling is fast, if frothing is fast, that will again lead to the adhesion of certain cells along the walls of the bioreactor, and these cells which adhere to the walls, they eventually lead to the formation of aggregates. Now such kind of problem can be avoided by using certain anti-foaming agents, or sometimes it also the problem with anti-foaming agents which have been observed over the period of time, that if they are being regularly used, they also reduce the oxygen transfer efficiency of the cells. So proper use of anti-foaming agents can prevent such kind of foaming or bubbling problem into the media. Fourth is the use of impellers into the bioreactors. Now impellers, we know that they are largely used for the proper mixing of the media throughout the culture, and this proper mixing is used to maintain the homogeneity and oxygen dispersion in the culture media. Now these media should have an ability, these impellers should have an ability to transfer the power over a larger volume of the bioreactors. And this ability to transfer the circulatory power across the media should be at a low impeller tip speed. It should not be that such kind of, or when we say ability to transfer the power, it's nothing but the ability to transfer the effect of the impeller to induce proper circulation across the culture bioreactor or the media that is present in the bioreactor. And if that ability to transfer the effect or mixing power into the bioreactor is achieved only at a high impeller tip speed, it will increase the shear stress into the culture media, thereby hampering the overall growth of the cells. Therefore, it is always recommended that the impellers, if they have ability to transfer the circulatory power throughout the culture media and the bioreactor, but it should be done at a lower impeller tip speed. And these impellers should cover a large surface area, or they should have a large surface area. Therefore, that will allow the transferring of that circulatory effect or circulatory power over the large volume of the bioreactor at a lower impeller tip speed, thereby preventing the excess of shear stress into the culture media and allowing the proper growth of the cells in the hairy root culture. Now here you can see these are some of the impellers that are used in bioreactors at commercial scale. So first one is a marine propeller, then we have a pedal propeller, anchor propeller, then bladed propeller, Rushton turbine propeller, spin propeller, helical and helical screw propellers. Now the name of all these propellers has been given on the basis of their structural characters, like Rushton turbine, it appears like a turbine. Then helical impellers, they move in a helical fashion. Helical screw, they again move in a helical helical manner, but they give more or less appearance like a screw. So all these impellers are given name based on their structural properties. Now it has been seen that in some of the bioreactors, because we know that bioreactors have propellers, they have different mixing mechanisms, and over the period of time such kind of impellers or mixing mechanisms, they lead to the development of certain amount of shear stress on the cells. Now to avoid this shear stress to impact the efficiencies of those cells for the producing secondary metabolites, we require certain support systems. Or in simple language, if I say, in order to protect those cells from the shear stress induced by the impellers or the bubbles or the mixers, we need certain support systems. And there are different support systems for different spray reactors or different type of bioreactors. Like a stainless steel separator is placed in the bioreactor in specifically in spray bioreactors, wherein the force of the mist of the nutrient that is prevented to affect the cells that are growing into the culture media. So there is a fine stainless steel separator is placed between the mist of the media and the cells that are present in the media. Or we can also introduce polyurethane foam or nylon mesh or baskets into the bioreactors that prevents the effect of impellers onto the growing cells in the culture media. Now these kind of polyurethane support systems or nylon mesh and basket support systems are largely used in stir tank bioreactors because they are using impellers to induce the mixing or circulation of the gases in the media. Now once we have taken all the precautions, we have taken care of all the problems that occur in bioreactors, next step is how you monitor the growth of those hairy roots, how you monitor the growth of the cells in the culture. Now there are different strategies by which we monitor the growth of hairy roots in the bioreactors. First one is the image analysis. Now this technique is largely considered as a non-invasive and non-destructive technique for monitoring the growth of the hairy roots, because you don't have to take the sample from the bioreactors, you don't have to open the bioreactors to take the samples, therefore it is considered as a non-invasive and non-destructive. Now there are different softwares that are being used for the image analysis, and these softwares are WinRhizo root scanning software or Root Edge software or RhizoScan softwares. Now these softwares they basically evaluate different parameters that are related to growth of the hairy root, and they help in evaluating the parameters like length of the roots, diameter and volume of the roots that is being growing in the bioreactor. They also help in calculating the total surface area that is covered by the hairy roots that are growing inside the bioreactor. They also help in assessing the branching point distribution. What are the points where the roots start branching? So it also helps in assessing those points and how that branching point is distributed across the culture media. Because if the oxygen circulation is not there, nutrient circulation is not proper in their culture media, as we discussed, that it will form certain dead pockets in the media, and these are those dead pockets where branching will be very less or will be negligible, and therefore this branching point distribution also helps in assessing whether there is a proper oxygen flow throughout the bioreactor, whether there is a proper nutrient flow throughout the bioreactor or not. Then how many root tips are being formed in the culture media. Then metabolite accumulation can also be assessed with the help of these softwares, because these softwares they evaluate the change in root morphology, they produce color images which also help in analyzing the metabolite accumulation. So such kind of parameters are used by these softwares in order to determine whether the growth in the bioreactor or the culture media is proper or not. Second technique or strategy is the using genetic algorithms. Now genetic algorithms, these involves a class of computational models, mathematical models, lying on the principle of natural selection and survival of the fittest phenomena. So if I translate into a simple language, then these are nothing but these are the mathematical models that help you in identifying the best variety or the lines of the hairy root cells that can grow properly into a media. Now for developing such kind of results, we take individuals from one population, they are selected on the basis of their fitness function, that is they are allowed to, or they are given more chances to reproduce, and they are allowed to form new population by application of natural selection operators. So we allow them to reproduce, to produce new generations, and the new generation or the offsprings that are produced, so by application of recombinant technology we introduce a genetic diversity. So first step is to select the best lines, we allow them to reproduce. Second is, once the offsprings are produced, we with the application of recombinant technology we introduce the desirable genes and we introduce genetic diversity. Now again these offsprings are again allowed to grow, and over the period different generations are produced, and this process is continued until a better population of individuals with improved characters over the parent populations is produced. So we can say we are just using best cell lines or best cells initially, allow them to reproduce, to produce generations We genetically modify those generations and we again allow them to grow till we reach a better population than the parent cells which we selected in the first step. So therefore, we can say that genetic algorithms are designed to assess the growth over the age, branching of the hairy roots, then the root lengths also. Third strategy is the use of artificial neural networks or ANN. Again, these are nothing but these are mathematical or computational models which mimics the structural and functional behavior of biological system. But the problem is we have to first give a certain set of input to these mathematical or computational models. So we can say that such kind of neural networks or computational models, they largely rely on the data which we put in them, and then they will give us the best output conditions for a particular culture media or particular combination of the conditions for the growth of the media. Now, it is a feasible method for modeling hairy root growth as well as culturing condition as we discussed. Suppose we are growing a particular hairy root culture in a given set of conditions and we get a certain set of results. Now, we put such kind of results into that mathematical or computational model, that on the basis of those results that we have put into that model, they will give a certain combination of those conditions which are best for our media. Now, we will grow our media with those permutation and the combination that have been given by the software, and from there we can make out which one is the best combination of the conditions required for the growth of the media. Now, it has also been found that in certain cases a combination of genetic algorithm and artificial neural network has been used, and this combination has been found to be better in comparison to when one of the strategies is used alone. Now, Mehrotra along with his group in 2008, they developed a model based on the combination of genetic algorithm and artificial neural network, and they named that model as feed forward back neuronal network. Similarly, Prakash and his group in 2010, they developed another regression and back propagation network wherein they predict the in vitro conditions for the optimum growth of the culture of Glycyrrhiza glabra. So this is how we can monitor the growth of the media, the growth of the cultures in the cells. We can either use imaging softwares, or we can use genetic algorithm, or we can use artificial neural networks, or we can use combination of genetic algorithm and artificial neural network. But problem with genetic algorithm is, or artificial neural network is, we have to put in certain data into these softwares, and then they will, depending upon that data, they will give us the output in terms of the best combination of conditions. So if your input data is biased, then obviously you are not going to get the genuine results of the combination of the conditions that are required for the cultural growth. So friends, in this lecture we discuss about the various factors that affect the overall growth of the hairy root culture in the bioreactor, starting from aggregates, adhesion, then proper aeration and mixing, then the impellers that are used. Then we discuss about the strategies by which we can determine the growth of the hairy root cultures, and eventually into genetic algorithms and artificial neuronal networks. So friends, in the next lecture we are going to discuss about the application of the hairy root cultures. Thank you.